So ladies and gentlemen, welcome. Today I will talk about oxygen and its role in metabolism. Again, you can find slides, so you can follow the slides, and I will talk about mostly transport of oxygen in the body. So it will be story of hemoglobin, then about electron transport chain, and a little bit about enzymes which use oxygen as a substrate, and about reactive oxygen species. And I will start with hemoglobin. So here you can see the structure in two different states. They are called the relaxed states and tense states. They look pretty similar, but if you switch to this video, maybe I can do it like that. Yeah, here we go. So if you look at the video, there is a visible change in the structure, and this change in the structure is crucial for the function of hemoglobin. So I will explain it in detail. So hemoglobin is a homotetramer. This is important. So it's composed of 4 identical subunits, 4 identical polypeptide chains. So it has a quaternary structure, and each chain, each subunit contains one binding site for oxygen, one molecule of heme, which you can see in these figures as these red spheres, and the secondary structure is the protein itself, and you can see that there are 4 different subunits like yellow, cyan, green, and magenta. And it can exist as a tetramer in 2 different states, R and T, which you can see in the picture. And in the absence of oxygen, I'm afraid this is swapped. Yeah, this should be other way around. I will check it to be on the safe side. In the absence of oxygen, it prefers tense state, and if it binds oxygen, it prefers the relaxed states. And there is a different affinity between these 2 states for oxygen, and as a result, the binding of oxygen to hemoglobin is not like the affinity, the strength of binding is not the same all the time for all concentrations of oxygen, but it changes with concentration of oxygen. I will show it on this slide. This is very important. So normally, if you have some protein, and this protein binds another protein or some small molecule, you obtain some kind of saturation curve. Naturally, at the 0 concentration of the ligand, there is 0 saturation, and at a very, very high concentration, there is 100% saturation. Something similar is also in enzyme kinetics, that enzymes, if they have no substrate, of course they do not catalyze any reaction, and if they have a very high concentration of substrate, they work at 100%. But usually this curve is like hyperbolic, so there is no this sigmoid shape. There is no this that goes like slowly up, then a little bit quicker, faster up, and then again slows down the increase. But it's like kind of monotonic. It's not hyperbolic. And this is, for example, the case of another oxygen binding protein, which is myoglobin, and this is a kind of counterpart of hemoglobin, but in muscle cells. So it's some intracellular binder of oxygen. And this protein simply binds oxygen with this saturation kinetics. So it's a hyperbolic. but hemoglobin has this sigmoid shape. So at very low concentration or at zero concentration, there is of course no oxygen bound, zero saturation, no oxygen, no binding of oxygen. That's clear. And if you increase oxygen concentration, usually oxygen or in general gases which can be dissolved in water, you can express their concentration as a partial pressure. It's also used in medicine that some concentration of oxygen or carbon dioxide in blood could be expressed in pressure units. So it's like you can imagine like if you take a blood and you put it into some container with a gas phase at the top, and you leave this system to equilibrate, and then you can measure the partial pressure of oxygen in this gaseous phase. So you can express concentration of oxygen in pressure, and you can somehow replace it by concentration in concentration units. So at very low concentration of oxygen, the binding is quite weak. So if you imagine like prolonging this curve in a hyperbolic manner, you would get something like very weak binding. But if you increase concentration of oxygen, then this binding becomes stronger. So this slope becomes higher, and then after, like if you get close to the saturation, you at some higher concentration you get close to saturation. And this is very important from the physiological point of view, because you can imagine that here at this point is like a concentration of oxygen, which is normally found in some organs, tissues. And in lungs, you can imagine like that the concentration is something like here. I hope the cursor is visible. So at around this O letter, for example, it's the concentration of oxygen in lungs. And here at this, when the slope goes slightly up, then there is a concentration of oxygen in some tissues, like for example in liver, let's say. And this is very important because in lungs the hemoglobin gets 100%, not 100%, but for example 90%. This looks like 90% here, so 90% saturated by oxygen. And then if the blood is delivered to liver, where concentration of oxygen is very low, then it gets like desaturated to just 10% of saturation. So like 80% of its capacity is used. If you imagine some hyperbolic shape, then there would be like very similar shape in this area for high concentration. So such a hemoglobin with normal saturation with a hyperbolic shape would get 80% or 90% saturated, but then in liver, for example, there would be saturation still like 40%. So it would go just from 90% to 40%. So it would work like some 80%, 50% of its capacity would be used for delivery of oxygen. So this oxygen would not be fully delivered to a liver, but would circulate through the bloodstream, and such oxygen would return to, or such oxygen and such hemoglobin would return back to lungs and would circulate around without efficiently delivering oxygen. If you imagine like prolonging this curve like this, then such a hemoglobin would be 50% saturated in lungs and 10% saturated in liver. So it would work again something like 40% of its capacity. So this sigmoid shape is very important and very useful for delivering oxygen, and it makes delivering of oxygen like very efficient. And this shape has been recognized by some measurements quite long time ago, and it was quite unclear why it works this way, because normal, like from physical chemical point of view, it's normal that binding of A with B gives this like saturation hyperbolic shape. And when this shape was explained for hemoglobin, it helped to elucidate the fact that there is something called allostery. Allostery means, or allosteric means like two different sites or different places in space, and it means that like binding, in this case binding of oxygen to one subunit, influences through the shape of the molecule binding of another oxygen to another subunit. So there is kind of distant distal regulation or distal influencing of one side by another side. And this became very interesting and very important for some other topics. So there are some other proteins, and for example enzymes, or some transport proteins, or some receptors which work in the similar way, that something binds to one side on a protein structure and it somehow causes some change in another side of the protein structure. And nowadays there is a lot of three-dimensional structures of proteins known. You can go to Protein Data Bank, which is a website where you can download one of more than 100,000 of three-dimensional structures of proteins, and you can look at the structure at the atomic level. And there are many examples there of some proteins which show some example of allostery, that some modification of one side of protein changes another side, another distant side of the protein, and this causes that the second side changes somehow its function, and it can activate some enzymatic or other activity. It can inhibit enzymatic or other activity, and it's very important in molecular biology. And it's possible to somehow show it in a three-dimensional space, on three-dimensional structures, how these changes take place. But in a time when this hemoglobin story was discovered and explained, there was very limited possibility to study three-dimensional structures of proteins, and this is quite nice example how such a change in structure was like indirectly explained, and indirectly there was some indirect proof of what's going on at the level of three-dimensional structure from simply biophysical measurements. So this hemoglobin changes structure a little bit with the as an effect of binding of oxygen. And at the time when this curve was known, but it was not explained, it was not clear what makes this shape here. It was a very nice example of how to explain some structural phenomenon simply based on some indirect measurements. It's still, I think this is still relevant because there are still some proteins, some of them quite important, for which determination of three-dimensional structure is quite difficult, and it's they are somehow resistant to some efforts to determine their three-dimensional structure. So we still cannot somehow explain everything by the three-dimensional structure, and it's sometimes quite good to look at such old-fashioned methods where people were able to learn a lot of things about three-dimensional structure of a protein from some indirect results. So here is the structure of heme. I missed here this R because or if you like, you can find the structure of heme like in full detail, and it's like porphyrin ring with iron 2+ in the center, and it's by coordination covalent bond, it's interacting with one of histidine residue in the structure of hemoglobin, and the opposite side of the iron atom is like ready for binding of oxygen. So with the oxygen, it is binding of oxygen take place here. It can also bind some other molecules which are mostly like toxic, and it can either deliver them to some other molecules for which they are even more toxic, like kyanite can be delivered by hemoglobin to different cells in which it blocks respiration chain, and that's the reason for high toxicity of kyanites. Or carbon dioxide blocks like hemoglobin, and it causes both like lack of delivery of oxygen, as well as like it delivers carbon dioxide to respiration chain when it's also makes some toxic effect. Methemoglobin is a hemoglobin in which this iron 2+ was oxidized to iron 3+. This may happen quite easily because the main job of hemoglobin is to carry oxygen, so it can easily oxidize. And in red blood cells, in erythrocytes, there are enzymes which can reduce this methemoglobin to normal hemoglobin. I will talk about it later when I go through the different biochemistries of different tissues and organs. So here is this very important shape of the curve. Normal binding protein has a hyperbolic shape without this kind of sigmoid shape. I would say that from the the best possible delivery of oxygen would be like a kind of stair function. Like at a low concentration there is 0 binding of oxygen, at a high concentration there is 100% binding of oxygen. So such a hemoglobin would like take 100% of its capacity, and this sigmoid shape is something like in this direction, but of course like this would not work from the physical chemical point of view. So like there is high high affinity at high concentration, so it gets almost 100% loaded, and low affinity at low concentration, so it easily releases all the oxygen at some tissues where there is a lack of oxygen. This behavior, of course, was very interesting, and the different people tried to somehow explain this, or to somehow make some models which can somehow explain this. And the first model was a Hill's model, and this model is not very realistic, because it assumes that the hemoglobin could be either like without oxygen or with or fully loaded with oxygen. So this is like a hemoglobin tetramer. Each of these 4 units are like individual subunits, and they could be either like without oxygen or fully loaded with oxygen. So all 4 binding sites are loaded with oxygen. If you look at this model and do some physical chemistry, like some thermodynamics, you end up with such a saturation curve. So this S is like saturation, could be 0 to 1. 0 means no oxygen bound, 1 means fully saturated with oxygen, no more oxygen can be bound. These square brackets are usually used in thermodynamics or kinetics, like to express concentrations of different components. So this, in this case, like this A is oxygen, and this Ks is a saturation constant. And normally, if there is some hyperbolic saturation, this kappa coefficients are equal to 1, so you can like simply remove them. And if you plot such a equation, this is constant, and how like saturation depends on concentration of some oxygen, for example, then you get a very similar curve, but nicely hyperbolic. And if you look at the concentration at which or if you set the concentration to this Ks, so here he would have Ks, and here we would have Ks. It's like Ks divided by 2Ks, so it's 1/2. So at the concentration equal to Ks, equal to saturation constant, you got 50% of saturation. This is like a standard binding of a ligand to a protein. But if you have in this Hill's model, instead of like 1, you got some kappa, and according to, because there are 4 different binding sites, this kappa should be 4. In fact, this kappa is not 4 for hemoglobin, so this model is not very accurate and not very realistic, but this somehow help to explain, and it makes it can make this kind of sigmoid shape. And still, like Hill's coefficient is still used, so to express this sigmoid character. So if Hill's coefficient is equal to 1, there is a normal like saturation with a hyperbolic shape, and if you increase Hill's coefficient, you change this more towards like this sigmoid shape. This model is not very realistic for one like kinetic reason, because it assumes that at one moment, like 1 molecule of hemoglobin with 4 molecules of oxygen will react, will somehow collide at one place, and will form like this complex, which is from the kinetic point of view very improbable that 5 different molecules like collide in proper way at the same time at the same place. But it somehow helped to explain the situation, and Hill's coefficient is used as a parameter to express the degree of allostery. The next model was Alder's model, which is somehow like not very, I would say, not very descriptive, because it simply assumes that like binding of the first oxygen happens with, here you can add some dissociation constant or some equilibrium constant, and this dissociation constant for binding of one oxygen is different from binding of the second oxygen and binding of the third and fourth oxygen. So at the low concentration there is low affinity, and so this dissociation constant would be quite high, and with the binding of more and more oxygen this affinity increases, so the binding becomes stronger and stronger. This is this can result to almost any value of kappa. It can also explain so-called negative allosteric effect, which means that binding decreases affinity, so the kappa then is lower than 1. But you need like some, in this case, 4 different equilibrium constant, 4 different dissociation constant. Here I have just 3, but here I missed one intermediate state. So 4 different constants are needed to make such a model, and you can imagine that like measurement, for example, of these constant by fitting this curve here is almost impossible, because you can fit almost any number there. But so then there was a very successful model by Monod, Wyman and Changé, and this looks at the first sight more complicated, but it is not. So in this case, they assume that the hemoglobin without oxygen exists as an equilibrium mixture, where there's equilibrium between the relaxed and the tense state. And I'm afraid here again it's swapped, so the tense state is more favored in the absence of oxygen. And both these forms can bind oxygen, but with a different affinity. So the relaxed state binds oxygen a little bit with a stronger affinity and then the tense state. So here is some dissociation constant for the relaxed state. Here is some dissociation constant for the tense state. And this constant here is the same as here, and same as here, and the same as here, and the same is like this tense state equilibrium or dissociation constant is same here, 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 and here. So we got like one constant, one dissociation constant for the relaxed state, one dissociation constant for the tense state, and we got some equilibrium between them. Or eventually it's possible to assume that the tense state doesn't bind oxygen at all. So we would have just 2 different equilibrium constant, and with this we can already get this sigmoid shape. So just how to explain it here. In absence of oxygen, there would be a dominant this tense state. But because this relaxed state binds oxygen more strongly, then if you add oxygen, you somehow shift this equilibrium towards the relaxed state. And if you add more and more oxygen, you more and more shift it towards the relaxed state. So at a low concentration where tense state is dominant, the binding is rather weak, because tense state affinity towards oxygen is low. But if you increase the concentration of oxygen as you somehow shift the equilibrium towards the relaxed state, the affinity is increased. And this model has helped to somehow explain how hemoglobin works. It works for these Hill's coefficient, which are not equal to the number of binding sites. So it can work for Hill's coefficient like between 2 and 3, which is found in hemoglobin. And finally, there is a model which I have the same picture as for this Alders model by Koshland, Nemeti, and Filmer. And this is like very similar to this Alder model, but there is one extra point, very important, which was like introduction to conformational change into this model. So Koshland is a quite famous name in connection with induced fit. So for example, some receptors, when they bind some ligand, they change the concentration, or some enzymes, when they bind a substrate, they change concentration, and this is known as induced fit. And the basis of this induced fit is in this model. So the story of hemoglobin in a time when there was very limited possibility to study three-dimensional structures of proteins somehow helped to explain that proteins are quite dynamic entities, and then they change their three-dimensional structure, and this change of three-dimensional structure has a quite important effect in their function. This is something which we now take as something natural and something trivial, but at a time when there was very limited possibility to study three-dimensional structures of proteins, it was quite pioneer. There were some pioneer works which explained this or highlighted this fact just from some indirect measurements. This hemoglobin affinity is not only influenced by oxygen, but also by some other changes. So, for example, change of iron 2+ to iron 3+ causes that the affinity of hemoglobin to oxygen is lost or almost lost. And as I mentioned, erythrocytes can use NADH to reduce iron 3+ back to iron 2+, and by this they can rescue hemoglobin. This change also, this oxidation of hemoglobin is also associated with a change in spectrum of hemoglobin, so it changes the color from like a red blood to more like brownish color. Carbon dioxide also influences binding of oxygen to hemoglobin, And this has also some kind of like a physiological role, because at low concentration of oxygen in tissues like liver or muscle cells or some tissues which consume some oxygen, they also produce carbon dioxide. And this carbon dioxide, together with acidity, carbon dioxide is like forms a little bit of carbonic acid, so it's acidic. So it changes the shape of this curve for hemoglobin, this saturation curve, and it somehow helps to like deliver oxygen in tissues more efficiently. And there is another like a compound which influences saturation of hemoglobin, and this is 2,3-bisphosphoglycerate. Maybe you know 1,3-bisphosphoglycerate, like intermediate of glycolysis. And in erythrocytes, in red blood cells, there is a glycolysis. Glycolysis is one of few metabolic pathways which take place in erythrocytes. And this glycolysis is slightly different, or there is some extra step or extra reactions, extra enzymes, which like form this 2,3-bisphosphoglycerate, and this 2,3-bisphosphoglycerate can influence the function of hemoglobin. So this is kind of like a linkage between the metabolism in erythrocytes, like glycolysis, and delivery of oxygen. So erythrocytes can use this to respond some concentration of oxygen. So erythrocytes and oxygen delivery and hemoglobin, it's very interesting story. In my opinion, the most important is that some people quite a long time ago, where there was a very few possibilities to study three-dimensional structures of proteins, they used such a simple experiments to learn something and to make some models which can explain behavior of hemoglobin, and by this they somehow replaced some structural biology experiments which we can do today, and they learned a lot of things about three-dimensional structures of proteins and dynamics of proteins without directly studying them. So now respiration chain, or electron transport chain, if you prefer this name. So this is a process which takes place in erythrocytes. Not erythrocytes, definitely not in erythrocytes, in mitochondria, sorry. And in mitochondria, there are two different membranes. One membrane, which is on the top, is kind of permeable. There are some pores in this membrane. And then there is the internal membrane, which is not permeable. And inside the mitochondria, there is space which is usually called a matrix of mitochondria. So, all the respiration chain is located on the this internal membrane of mitochondria. This outer membrane of mitochondria has very little role in respiration chain. It can easily, protons can easily pass through this membrane, because there are some pores. Only molecule which is somehow confined in this intermembrane space is cytochrome C. It was found later that cytochrome C is also important as a signaling molecule, and it can signal some damage of mitochondria, that if mitochondria are damaged, they can, like this outer membrane is damaged, they can release cytochrome C, and cytochrome C can do some signaling job inside the cell. So inside mitochondria there are different processes taking place, like a Krebs cycle, or if you prefer citric acid cycle, or tricarboxylic acid cycle, or fatty acid degradation, and these processes are source of NADH. So they permanently reduce NAD plus to NADH. And a respiration chain somehow converts this NADH back to NAD plus, or there is some extra input, and at the end, like these electrons which are taken from this NADH, they go through this electron transport chain or respiratory chain to this final molecule cytochrome C oxidase, and the final acceptor of these electrons is oxygen, molecule oxygen, O2, and this oxygen is reduced to water. And very important is that during this process there are many like protons being either released from some chemical reaction, or more importantly somehow like the energy released by this reaction is used to pump these protons across the membrane, which forms some concentration gradient on the membrane, and this concentration gradient is then used as a driving force for production of ATP. And when I started to study biochemistry some long time ago, in most of textbooks of or in all textbooks of biochemistry that time, there were some pictures like that, with the exception that it was not possible to see here like three-dimensional structures with some alpha helices and some structural elements. But all these like molecules, all these complexes, membrane complexes, were shown as some kind of like balls or some symbols, and there was very limited knowledge of their three-dimensional structures. So they are membrane proteins, so like a determination of a three-dimensional structure of a membrane protein is a bit like more complicated, and it's not very trivial. So it took some time to decipher the three-dimensional structures of all these complexes, but now they are completely known, so you can look at these structures at the atomic level, and you can watch the pathway of electrons, for example, or you can look at the active site, look how they interact with membrane, and many other things can be found from these three-dimensional structures. So here is the complex 1, and the complex 1 catalyzes the reaction in which NADH reacts with ubiquinone, and it forms NAD plus ubiquinol, or ubiquinone H2, or the reduced form of ubiquinone. I will show later the structure. So you will see how this ubiquinone and ubiquinol looks like, and what is the difference. So there is an active site for this reaction of which can be like this reaction which take place somewhere here is like NADH gives NAD plus, plus H plus 2 electrons. So this is reaction which happens somewhere here, and these electrons are being transferred from this point to somewhere here, where is the second active site. How electrons can go through the space? So normally if you have some electric cable, there is a metal which through which electrons can pass, and it's something similar here. So there are some centers, like very common are iron sulfur clusters. I will show some structure or some schematic view. So there are some like moieties here where iron atoms are being like bound by some sulfur atoms, and this is covalently attached to the protein structure. This iron can exist in 2+ or 3+ states. So if it's in 3+ state and it accepts electron, it goes to 2+ state, and then it can release electron to another site. So this is similar like what happens if you have some piece of iron and you somehow put some electric voltage on it, and the electric current goes through this piece of iron. So this is something similar here. So these electrons go from this side to another side, and in this another side there is the reaction in which ubiquinone reacts with protons and with these electrons, which are delivered by this electron relay, and the reduced form of ubiquinone is formed. This ubiquinone, I will show the structure in a moment. So it's a molecule which can be somehow like dissolved in the membrane. So here there is everywhere water, and there is everywhere water, but here is a membrane, and inside the membrane there are fatty acids chains which form the structure of membrane, and this ubiquinone can be easily somehow dissolved in this non-polar environment of the membrane. I would say that ubiquinone is kind of counterpart of NAD+. So NAD+ can exist in oxidized form and the reduced form. So in one reaction, like one of Krebs cycle reaction, for example, isocitrate dehydrogenase converts isocitrate to 2-oxoglutarate, and NAD+ goes to NADH, and in another reaction catalyzed by complex 1, this NADH goes back to NAD+. So it's typical like coenzyme, and ubiquinone is also a coenzyme. So in this reaction it goes from the oxidized state to the reduced state, and then it can somehow float through this membrane until it finds another complex. It will be complex 3. I will show the reaction in a moment. And in this reaction it is oxidized back to ubiquinone. The main difference between NAD+ and ubiquinone is that NAD+ is like soluble coenzyme, so it occupies, or it can float anywhere in this three-dimensional space of mitochondrial matrix, and of course it can be also in the cytoplasm of the cell or in the intermembrane space as well. But this ubiquinone is confined in this two-dimensional space of the membrane, so it can be like oxidized by one enzyme here and reduced by another enzyme here, and between this it can somehow jump between different these complexes, but only in this two-dimensional space of the membrane. So here is another complex 2, succinate dehydrogenase. Sorry, here added here some systematic enzyme names and enzyme classification codes. So sorry for being this somehow hidden. So it's the reaction is that succinate with ubiquinone forms fumarate and reduced ubiquinone. So the reaction will be succinate. Then it's colon, ubiquinone, and oxidoreductase is a systematic name. Of course, you don't have to know the systematic name, so it doesn't matter that it's hidden. What is interesting in this enzyme is that this is like a reaction from Krebs cycle. This is like a direct attachment between the respiration chain and Krebs cycle. So this is one of reaction of Krebs cycle in which succinate goes to fumarate, and here there is a prosthetic group, a piece of enzyme which is not of protein character and which is attached to the enzyme, and this prosthetic group called FAD, flavine adenine dinucleotide. I will talk about coenzymes and prosthetic groups next week. So this FAD goes from the oxidized state to the reduced state. Then it releases protons, and these electrons go again through some kind of rally. Here they jump from one center to another, and at the end there is again some reaction in which ubiquinone is reduced to ubiquinol. So this ubiquinol can come either from the complex 1 or from the complex 2, and it ends at the complex 3. So the complex 3 takes this ubiquinol, or in the systematic name it's quinol, and cytochrome C, in the systematic name it's ferricytochrome C. And this cytochrome C gets from the this reduced form ferricytochrome C to the oxidized form ferrocytochrome C. Sorry, from the oxidized form to the reduced form. Sorry, yeah. So the cytochrome C, I will show in a moment the structure. It's a small protein. Maybe it's somewhere here, like maybe this or this. I don't know exactly. And it's a small protein which binds iron, and again iron can exist either in a 2+ or 3+ states. So here because it's reduced, then this will be in 3+, and then it's oxidized, so this will be in 2+ state. And finally, this cytochrome C delivers electrons to cytochrome C oxidase, which is the complex 4, and this complex 4 oxidizes this cytochrome C back to its oxidized form, and it oxidizes this by molecular oxygen, and it forms water. It's quite big multi chain protein, and it's probably one of most important protein, because without this it's not possible to breathe. That there are also many other proteins which are key for breathing, but this is maybe the most important one. I promised to show the structure of coenzyme Q, ubiquinone. So here is the structure of coenzyme Q. There is a chain which can be a variable in length, so it could be 6 to 10 these repeating units. And if you look at this molecule at the first sight, so imagine that this is like that you draw it like explicitly, then this molecule is highly hydrophobic. So it's there is very low solubility in water, and it will be dissolved in a membrane, and it will travel through the membrane in this two-dimensional space. And this is the oxidized form. If you want a reduced form, you have to add hydrogen to this oxygen here, and this oxygen here. So these double bonds will change to a single bond, and here there will be like aromatic rings, so like conjugated double bonds. And cytochrome C is a small protein, so if you compare with these monsters, I did my best to like plot it in the same unit, so it's same scale, so it's like a very small protein with a iron bound on a heme. And again, it can exist, this iron can exist in 2+ or 3+ state. As I mentioned, the cytochrome C can have also like some signaling role, which is quite independent on its electron delivery role. So when some mitochondria are damaged, the cytochrome C can be released to the cell, and it can do some signaling, like some, for example, in apoptosis, this is one of signaling molecule in programmed cell death. Not so long time ago, it was found that this respiration chain can form such a kind of mega complex, that these players of the respiration chain are not fully like separated, like this picture which I've showed at the first slide. This is not very accurate, so it's not like that they are such a separated, but they actively interact with each other, and they form such a kind of mega complex. And this mega complex makes this life easier for all these cofactors to somehow like deliver electrons. So if you like, you can go to a protein data bank where you can find this structure. I don't know the code at the moment, but I think it's quite easy to find it as a respiratory chain megacomplex or electron transport chain megacomplex. And there is quite a recent three-dimensional structure determined by cryo-electron microscopy. I don't know exactly how this is embedded in a membrane, but I think there is some kind of this is like a membrane interface. So if like a membrane is in like this in the plane of the slide, little bit like shifted towards us. Yeah, so you can imagine how it's embedded in the membrane. So here is some kind of overview how it works. So this is the first scenario in which these electrons come from NADH. So NADH goes to NAD plus to plus H plus, and electrons which are released, you got here 0 charge, you got here 2+ charges. So there must be 2 electrons released, and they travel by this electron relay to another site, in which coenzyme Q plus 2+ gives the reduced coenzyme Q. And this reaction releases energy, and this complex 1 is so sophisticated, so clever, that it does not change this energy to heat, but it uses this energy to pump protons across the membrane. And so 4 protons are being transported across the membrane. And this is like active transport, so here some proton binds somewhere here, and then some conformational change happens, then it cannot get back and must go this way, and This happens like 4 times, so 4 electrons are being transported. Then this coenzyme Q in the reduced form, this ubichinol, goes to complex 3, where it's oxidized. 2 protons are released in this step, and they are released to this side of the membrane. So they came from here and they were released here, so they are also like transported. And at the same time, 2 protons are being again like transported by an active transport. So again, these 2 protons bind somewhere here, then there is some conformational change, and then they have to go that way. And electrons which are released in this process, they go to cytochrome C. So cytochrome C gets changes from oxidized to reduced. Sorry for the either swap this or swap the arrows, sorry. And this cytochrome C delivers electrons to the complex number 4, which is cytochrome C oxidase. And cytochrome C oxidase takes these electrons, and with protons it forms from protons, electrons and oxygen, it forms water. And also at this point, 2 protons are being actively transported. And here it should be like that 8 protons at the end should be like transported. There is some proton being produced here and some proton being taken here. So if you take it over and over, there should be 4 protons transported. There is 8 protons transported. There is an alternative entrance to the respiration chain, which is from the Krebs cycle. So in Krebs cycle, when succinate is being transformed to fumarate, this releases 2 protons and 2 electrons. So at the beginning there is FAD prosthetic group converted to FADH2, and then this FADH2 releases these protons, and electrons travel here and they convert like a coenzyme Q from oxidized to reduced state, and then the rest is the same story as in the previous slide. The result of this is that like only 4 protons are being transported. So here should be like here we got 6, and here we got this is 6, but you have to subtract these 2 protons, so at the end 4 protons are being transported. And all this results to some high concentration of protons on this side of membrane and low concentration of protons at this side of membrane. And this is the driving force of ATP synthesis. So there is an enzyme called ATP synthase. Here is the very nice structure, which allows protons to go back across the membrane to return. And this energy which is released by this process is used to produce ATP from ADP and inorganic phosphate. This so-called proton motive force has like 2 different components. First, there is a concentration gradient. So there is a higher concentration of protons here than here. And another component of this proton motive force is because if you look at this slide or this slide, it's really like that hydrogens, like protons are being transported across the membrane. So it's really that like for protons or maybe oxonium cations are taken from here and put here. And there is no like co-transport of some chloride or other counter anions. So you take some particles with plus charge from one side of membrane and you put them to another side of membrane, and you do not do anything with any like minus charged particles. So at the end there is an excess of plus charged particles of protons on one side and lack of concentration of plus charged particles on the other side. So this is also like this also forms an electric potential. So this is the second component of the proton motive force. And this is used as a driving force for synthesis of ATP from ADP and inorganic phosphate. And it was found that this molecule, I will try to, I will try to switch on the movie. Doesn't work. So it was found that this molecule rotates when it's doing this job. So it was at the beginning some kind of like kind of theory. It was not clear whether it rotates or not, and some group which somehow proved experimentally that ATP synthase indeed rotates. What they did, they first took first they isolated ATP synthase from mitochondria, and they somehow broke this membrane. It's possible to do it by some detergents. It's necessary to use some mild detergents, otherwise you would break the three-dimensional structure of protein. So they used some mild detergents to somehow like remove the ATP synthase from the membrane. So what they get was some like molecules of ATP synthase in solution, in which they were in this part which normally is embedded in the membrane, they were like somehow coated by some detergents. And such a isolated ATP synthase cannot do its job because there is no proton motive force, because this and this space is the same, and any protons can easily like diffuse if there is no membrane. But what these enzymes can do is like to catalyze a reaction in opposite direction. So if this isolated ATP synthase is loaded with a high concentration of ATP, it can catalyze the reaction in opposite direction. So it can decompose ATP to ADP and inorganic phosphate, and at this reaction it rotates, but in opposite direction. And this rotation is used to pump protons in like opposite direction, but it doesn't have any effect because these protons which are being pumped from this side to this side can easily return in case of absence of this membrane. So when you somehow dissect ATP synthase from membrane and you put it into solution, it still can work. It can decompose ATP to ADP and inorganic phosphate, so it can do the reaction in opposite direction. It can also somehow oppositely pump protons, but this pumping has no effect because these protons can return. And what they did, they put here, using some genetic engineering, they put here some kind of like a moiety, some extra polypeptide chain, and by this polypeptide chain it was possible to attach this complex onto some slide of under microscope. So they like attach several of these molecules on the microscopic slide, and then what they did, by some covalent linkage, they took actin chain. Actin is a protein which is a part of cytoskeleton and also important in muscle work, but they use it because it's simply some long protein, so they attached a long protein chain somewhere here. They covalently bound it to this molecule, and this actin was previously fluorescent labeled. So when this molecule is very long, it's quite relatively easy under microscope to see that it's moving. So what they did, they prepared such an assembly where this ATP synthase was like dissected from membrane, was attached on a microscopic slide, and then they attached this fluorescently labeled actin on it. And then when this molecule was loaded with ATP, this part started to rotate, also the actin started to rotate, and it was possible to visualize it using microscope, and this is what you saw in the video. And this rotation is like dependent on concentration of ATP, so if they add more ATP, it rotates more. If they add a lower concentration of ATP, it rotates slowly, and if there is no ATP, this rotation is absent. So this is quite elegant experiment to show that like ATP synthase rotates. There were also other like quite elegant experiments to decipher how this respiratory chain works. Generally, if you want to somehow study some metabolic process, you can try to isolate individual enzymes, and you can try to somehow characterize their reactions. And also as an alternative way, it's possible to use some inhibitors of enzymes to show that if you block certain enzyme by an inhibitor, then all these intermediates of metabolic pathway which precede this enzyme, all the substrates and all the intermediates like before these enzymes, they start to like accumulate because the enzyme is blocked and they cannot be converted through the metabolic pathway, and all the products and all the like intermediates after this enzyme, their concentration would go down. So this is a quite useful tool or strategy how to study some metabolic pathway by trying to block certain enzyme and measuring concentrations of different intermediates and what happens. And this was also used to find how respiration chain works. So for example, cytochrome oxidase enzyme, the complex number 4, can be blocked by kyanide, by carbon monoxide, or by sulfane, or by azide, and all these like toxic compounds, and they are toxic because mainly because they block this enzyme, and these toxic compounds played a role in deciphering how the respiratory chain works. This chemiosmotic theory, which means the theory that proton motive force is formed by respiration chain, and this proton motive force is then used to synthesize ATP, has been quite controversial for quite a long time. I would say it's one of last metabolic pathway that was deciphered in at least in human metabolism. And there are again some experiments and some compounds which helped to show that or used as evidence or as a kind of indices that it works in this way. And very important compounds are so-called decouplers, and decouplers are compounds which can somehow kill proton motive force. One of them is dinitrophenol, and this is a compound. If you imagine the structure of like phenol, there is OH group on a aromatic ring. It's bit acidic, or this one is quite strong acid, but it's still like quite non-polar. So this compound can easily pass through the membrane in both forms, like in protonated and deprotonated form. So if there is a higher concentration of protons on one side of membrane, this molecule can get to this side of membrane in deprotonated form, and then it can take protons, take them through the membrane and release them on the other side of the membrane, and by this it can kill the proton motive force. And this works, and this was one of argument for this proton motive force, and from this chemiosmotic theory that a respiration chain forms proton motive force, and proton motive force in turn is a driving force for ATP synthesis. Another molecule which is also quite interesting is an antibiotic called valinomycin, and it is a peptide which can make a pore through the mitochondrial membrane, but this pore can only potassium cations can pass through this pore, not protons. And what happens if you add valinomycin to some mitochondria which are actively metabolizing? So this valinomycin forms pores into a membrane, and only potassium cations can pass. And if there is a high concentration of protons on one side of the membrane and low concentration of protons and at another side of the membrane, but these protons on one side of membrane would have some counter anions, then nothing would happen. But if there is a high potential, electrostatic potential on the membrane, which means that there is a excess of positively charged particles on one side of membrane and lack of positively charged particles on another side of membrane, and there is some potassium cations at the same concentration on both sides of the membrane, it happens that these potassium cations will go through the valinomycin pores to another side of the membrane to somehow compensate this gradient of charges. So this valinomycin does not uh, influence concentration gradient of protons, but it influences and it somehow seizes, or it somehow blocks charge gradient, so electrostatic potential. So by this using this valinomycin, it was possible to show that this proton motive force has these two components. One is the concentration gradient of protons, and the second one is the electrostatic potential, or the gradient of charged particles. And quite interesting is that there is a protein called thermogenin, which is expressed in some animals which undergo some hibernation in winter time, or also in human newborn infants. And this thermogenin can, when it's present in mitochondria, it can simply make some pore in a mitochondrial membrane, and it can allow protons to go back without producing ATP. And so this energy, which is stored in proton motive force, is then converted to heat. It's kind of dissipated, and in this case it's useful because it can somehow increase the temperature. So for some hibernating animal, it can help them to somehow wake up, and it can be also useful for newborn infants to heat themselves up. So at the last two slides I would go through enzymes which use oxygen as substrate, and also reactive oxygen species. So there are two types of enzymes which use oxygen as a substrate. They could be found in human metabolism as well as in some other metabolisms, and I will present here some enzymes which are quite interesting from the point of human metabolism, as well as some enzymes which are interesting from the point of, for example, some analytical purposes or some biotechnologies. So there are enzymes called oxidases, which oxidize a substrate by a molecule of oxygen, by O2, but they do not incorporate the oxygen into the product. So we already had complex number 4, cytochrome C oxidase. So it takes oxygen, cytochrome C in the reduced form. The product is cytochrome C in oxidized form, and water, and also protons are needed for this. But this oxygen, this O2, ends up as water, and it's not incorporated, or it's doesn't end up on cytochrome C. So that's why it's oxidase. And I also mentioned here two oxidases which are not human enzymes, and they are interesting from the point of some biotechnologies, or especially from the point of view of diagnostics, clinical biochemistry. So if you go to doctor and they take a sample of your blood, and they order some analysis of different biochemical parameters of the blood. One of them could be glucose concentration, and one of them could be cholesterol concentration. And for measurement of glucose concentration, the enzyme called glucose oxidase is usually used. This enzyme oxidizes glucose to gluconolactone using molecule oxygen, and oxygen is reduced to hydrogen peroxide. It's enzyme which is fungal. It's mostly from some penicillium species, and this measurement of concentration of hydrogen peroxide is quite easy. There are many different ways how to measure hydrogen peroxide, or how to measure increase of concentration of oxygen. So it's very often used in diagnostics to measure concentration of glucose. It can be either done by some spectrophotometric measurement or in some sensors. For example, there is a glucose sensor used either in clinical laboratories or people who suffer diabetes. They often measure themselves the concentration of glucose in blood by taking a drop of blood and putting it onto some device where there is a like electrode, and this electrode is somehow coated or contains immobilized this glucose oxidase enzyme to measure concentration of glucose. I will talk about this later in clinical biochemistry lecture. So I will explain how these assays are done in detail. And there is similar story of cholesterol oxidase. So if you want to measure cholesterol in blood, then there is a microbial enzyme which oxidizes cholesterol. Again, it produces hydrogen peroxide, and again, hydrogen peroxide can be used, can be easily measured. And there is high density and low density lipoprotein, and I will talk about it later. So these are often referred to as good and bad cholesterol. So it's like particles in blood which contain some lipids, cholesterol, and some proteins. And usually these high density lipoproteins and low density lipoproteins are determined as a cholesterol which is present in different types of particles. So also measurement of cholesterol is used or uses or measurement of this high density lipoprotein and low density lipoprotein is based on measurement of concentration of cholesterol. The second type of enzymes which use molecular oxygen as substrate are oxygenases. So they also oxidize a substrate by a molecular oxygen, but unlike oxidases, they incorporate oxygen atoms from O2 into the product. There are so-called monooxygenases which incorporate just one of them, and dioxygenases which incorporate both of them. Here I mention cyclooxygenase, which is a human enzyme which is involved in inflammation. The reaction is pretty complicated, the name is pretty complicated, so I will not go through details. It's important that it's a target of aspirin, ibuprofen, diclofenac, and many other like drugs which are used to treat fever, pain, inflammation. Another interesting enzyme is the procollagen proline dioxygenase, which oxidizes collagen, and it's responsible for like proper structure of collagen, which is the like protein present in different like connective tissues, and this is the one of reason why we need vitamin C, and the lack of vitamin C results to some changes in procollagen or changes in collagen structure, and this is because this requires iron 2+ for its catalytic activity, and there is ascorbate, so vitamin C used to somehow keep this iron 2+ in oxidized in the reduced state. Otherwise this enzyme doesn't work. So there is this procollagen, which is like a chain of polypeptide chain with high concentration of proline, and these prolines have to be hydroxylated, like post-translationally modified, and this is reaction with oxygen. So one of atoms of molecular oxygen will go to this procollagen to form this hydroxyl group on proline, and the second oxygen will oxidize to oxoglutarate, to succinate and carbon dioxide. And another example of oxygenases is cytochrome P450. This is quite large group of enzymes which are responsible for detoxification in human body, and I will present them later. I will somehow mention them because these are liver enzymes. So when I talk about liver, I will present cytochrome P450. And in the last slide, let me briefly mention reactive oxygen species. So there is something called oxidation stress, because like a human organism is aerobic. It uses oxygen as oxidating agent, and it may happen during these processes some particles like hydrogen peroxide or superoxide or hydroxyl radical or singlet oxygen can be formed. For example, hydrogen peroxide can be toxic because it damages membrane structures, it can damage DNA, and it can damage some other components. So cells have developed some tools how to fight with these reactive oxygen species. In animals and in some bacteria, hydrogen peroxide is usually being decomposed by the enzyme called catalase. And the reaction is that two molecules of hydrogen peroxide react, and they form two molecules of water and molecule of oxygen. So hydrogen peroxide is easily decomposed to molecule of oxygen by an enzyme called catalase. In some countries, hydrogen peroxide is used as a kind of antiseptic agent. So if you get some scratch on your skin, then it's possible to apply some 3% solution of hydrogen peroxide. And if you have ever done this hydrogen peroxide solution starts to bubble. And this is because the presence of the enzyme called catalase in these cells. So it's very efficient enzyme because these enzymes are ready to decompose any hydrogen peroxide which can be formed in a cell. Plants have quite a different strategy. Plants usually produce a lot of different compounds, like some phenolic compounds, different antioxidants, we would say, there are different secondary metabolites produced by plants, and many of them are reducing agents, so they can be used to reduce hydrogen peroxide. And many plants have an enzyme called peroxidase for this purpose. So the reaction catalyzed by peroxidase is a reaction of hydrogen peroxide with some reduced form of some compounds, and it produces an oxidized form of some compounds. The enzyme called peroxidase is also very popular in many different applications in clinical biochemistry, in some other or in as a labeling enzyme, because there were especially the enzyme called enzyme from horseradish, isolated from horseradish, is very often used. Horseradish peroxidase, there are many synthetic compounds which were developed in a way that these compounds are colorless, and when they are oxidized by hydrogen peroxide under catalysis of peroxidase, they produce some color product. So this can be either used to measure concentration of hydrogen peroxide. For example, by this you can measure concentration of hydrogen peroxide which was produced from glucose by glucose oxidase. So this can be the second enzyme used in measurements of glucose in blood. Or it can be also used as a kind of labeling enzyme, so you can attach peroxidase to, for example, some antibody, and then, for example, under microscope you can visualize which part of tissue interacts with antibody using this peroxidase. So it's very, very interesting and important enzyme. Superoxide radical, so it's like oxygen with charge sorry, here is a typo. It should be like 2 should be as a lower script, so it's like oxygen with like 2 atoms minus charge, and it's at the same time radical. So superoxide radical can be decomposed by an enzyme called superoxide dismutase. So it takes like 2 superoxide ions, and it forms oxygen from one of them and peroxide from the second one of them. And it's again a very efficient enzyme. It was at one moment supposed to be like a good target for anti-cancer therapy, because cancer cells produce a lot of these oxidative, this reactive oxidation species, and it was supposed that by blocking superoxide dismutase, it would be possible to somehow make bigger damage to cancer cells compared to normal cells, healthy cells. But as far as I know, it's not, this concept has not been somehow used in clinical practice. And there are some other like reactive species like hydroxyl radical or singlet oxygen. Singlet oxygen is just O2 but with some different spin arrangement, and it's more reactive than regular oxygen. There is a quite important like way how cells cope with oxidation stress, and this is a peptide called glutathione. Glutathione is composed of 3 amino acids, is gamma-glutamyl cysteine glycine, and this cysteine can form a disulfide bridge. So in a reduced form, this side chain is like normal with SH group, but when it's oxidized, it forms a dimer with a disulfide bridge. So there are 2 these peptides connected with a disulfide bridge, and this reduced form with SH group can react with all these molecules and can be converted to this oxidized form, and it can be reduced back by NADPH, and therefore we can see NADPH as a kind of main antioxidant in a cell. So that's all for today, and see you next week with the cofactors, prosthetic groups, and other components of enzymes.